So now we will start with C4 cycle, which is also known as the Hatch and Slack pathway. Let us talk about that. So Kortchak and Hatch, they first observed that there are 4 carbon compounds, that's OAA, the oxaloacetic acid, which is formed during the dark reaction in the sugarcane leaves, right? Now, Hatch and Slack in 1967 studied in detail and proposed a pathway for this dark reaction in the sugarcane and the maize leaves. So they found that the first stable product of this reaction is OAA, oxaloacetic acid, which is a 4 carbon compound and a dicarboxylic substance, and thus Hatch and Slack pathway is called as C4, or we call it as dicarboxylic acid cycle, DCA. Now you could see this diagrammatic representation of the Hatch and Slack cycle. You could see that this is the zone, this is the area of the mesophyll cells, and this is the bundle sheath cells, and probably this is the area where most of the photosynthesis takes place. Now you can very clearly see that how the phosphophenol pyruvate fixation takes place, regeneration of the C3 acid, C4 acid, and C4 decarboxylation is taking place, and this cycle you could see over here. The C4 cycle occurs in 1500 species of 19 families of the angiosperms, but most of the plants, they are monocots, and which belongs to the family Gramineae, the grass family, Poaceae and Cyperaceae. That's sugarcane, maize, sorghum, oats, chloris, then sedges, bajra, panicum, allotropis, all of that, right? Now, talking about the dicots with C4 cycle, that's Euphorbia species, Amaranthus, Chinopodium, Bravia, Atriplex, Rosea portulaca, Tribulus, all of that. Crans with anatomy present in the leaves of the C4 plants. This is, let's just get into this detail. So what exactly you see in here is, you must be aware of this diagram. That's the stoma and the veins and the lower epidermis. You could see the upper epidermis, the bundle sheath shells with large chloroplast, the mesophyll cells with few small chloroplasts that you can see over here. So since you are aware of this region now, and here comes the question. The question is, in leaves of the C4 plants, the malic acid synthesis during CO2 fixation occurs in where? And the options are epidermal cells, mesophyll cells, bundle sheath cells, or is it guard cells? What do you think is the answer? And the answer here is, it's a B, that's the mesophyll cells. Of course, the malic acid synthesis will definitely take place in the mesophyll cells, and that's the reason why I showed you the diagram. Now, talking about the cran's anatomy, there are various features of the cran's anatomy that we need to know. For example, the green bundle sheath cells, the BS, the bundle sheath cells, they are present around the vascular bundles. So you could see, if you try to compare the C3 cycle with the C4, that's the cran's leaf anatomy, you could very clearly see this is C3 rice, this is C4 maize. So in C3, what do you see? You see the wide vein spacing, single photosynthetic cell type, and here this is a closed vein that is spacing 2 photosynthetic cell type, bundle sheath and the mesophyll. Right? Now there are various features that we are going to talk about. The second feature is dimorphic chloroplast. So here this is an interesting fact that there is a dimorphic chloroplast which are present in the leaf cells. So yeah, so these are some dimorphic chloroplast. Apart from that, the dimorphic chloroplast, they are present. The chloroplast of the bundle sheath cells or the gran cells, they are large and they lack the grana. And the mesophyll chloroplast, they are small and they do have the grana. Now talking explicitly here over the C4 cycle, there is a Hatch and Slack pathway. In C4 cycle or in C4 or the C3, these cycles, they occur in the bundle sheath. You must be aware of this. While the C4 occurs in the mesophyll cycle. Okay. So C4 in the mesophyll and C3 in the bundle sheath. So this is C3 mesophyll cycle, and this is the mesophyll cell, and here C4 you see in the mesophyll as well as in the bundle sheath. Now operation of the Hatch-Slack pathway requires cooperation of both the photosynthetic cells, that is the mesophyll cells as well as the bundle sheath cells. Both have to cooperate well. And also, photosynthetically, when we talk about the C4 cells, they are more efficient and there is no Warburg effect, or we call as the photorespiration, okay, in C4 plants. The reason is that the site of rubisco in the bundle sheath cells, that has less oxygen, it's present there, so less oxygen is present there, and mesophyll cells, they pump more carbon dioxide for the C3 cycle. Now, C4 plants, they are found in the tropical habitat. They have adapted themselves for the higher temperatures, low water availability, also intense light. So they are adapted to these conditions. This is a C3 plant. This is a C4 plant. You could very clearly identify the different parts. You could see there's upper epidermis, the mesophyll area, these are the palisade cells. You could see the bundle sheath cell here, and here you could see here lower epidermis, the stoma, and you can clearly differentiate between the C3 and the C4. As a result, the C4 plants, they are more developed and they are more adapted than the C3 plants. They do not cause any loss of photorespiration. Now that is an adaptation, that is an adjustment, that is some form of evolution that they see, that they show, that they practice. So this is the C3 plants. You could see the bundle sheath, the mesophyll area, vein and stoma, and here you see here the C4 plants, and you could see the stoma, you could see the bundle sheath cells, you could see the mesophyll area. Yeah. Now, talking about with respect to oxygen, in a concentration of oxygen increases artificially, and then the photorespiration may be started in the C4 plants. Now, first carboxylation in the C4 cycle occurs by the PEBcase in the mesophyll chloroplast, while the second carboxylation or the final carbon dioxide fixation by the C3 cycle occurs in the bundle sheath cells. So you see this is a bundle sheath cells. So this is C4 cycle, and here the bundle sheath chloroplast.